Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Wyatt Technology Corp., I'd like to welcome you to high-throughput analytics for formulation and process development of bio and nanotherapeutics. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. Now, I'd like to introduce our presenter for today. He is Dr. Daniel Sohm, PhD, Principal Scientist for Wyatt Technology Corp. Wyatt Technology is the recognized leader in light scattering instrumentation and software for the determining of absolute molar mass, size, charge, and interactions of macromolecules and nanoparticles in solution. Wyatt's innovative products have been cited in over 12,000 scientific articles and are relied upon by leading scientists in these fields around the globe. So with that, welcome, Dr. Sohm. The presenter ball is yours.
Hello, Elizabeth, and thank you for that introduction, and welcome to everyone joining in today. As Elizabeth mentioned, it is my pleasure to discuss with you high-throughput tools that Wyatt Technology offers utilizing light scattering to accomplish the tasks that you are faced with in the course of formulating and developing processes for biotherapeutics and nanotherapeutics. And so in the course of those development efforts, you will run through several stages. And the first is, given that you start with various candidate molecules or particular conditions to manufacture the nanoparticles that are being used as a therapeutic, whether they're biologics or whether they're small molecules which are formulated in delivery systems such as a liposome or other nanoparticle, you're all going to go through the same essential steps, which are, given what we have, are we on the right track? Do we have the right candidate molecules or the right conditions from which we can select the few which are the most promising? And in the course of those evaluations, you'll be called upon to measure the size. Does your sample aggregate? How much will it aggregate? What is propensity for aggregation? How stable is it in terms of thermal properties and colloidal properties? And if your sample happens to be a high concentration monoclonal antibody, you'll also be looking at its viscosity, which is a critical issue for those types of therapeutics. Once you've selected a few of the candidates or conditions, you'll then go on to test them more deeply, for instance, by applying stress or looking at shelf life stability studies. And throughout those measurements, typically a few times during the course of the stability course and at the end as well, you're going to do a deeper characterization. So for instance, measuring the size and molecular weight, the types of aggregates are forming, are there any fragments, other subtle modifications that could be reflected in charge or particle-particle interactions. And in the course of an intelligent development process, you're also analyzing along the way, why are you successful or why does a particular candidate or condition fail? And for that, you need information about the intermolecular or interparticle interactions that affect the formulation or affect the process conditions, and things like self-association of the molecule, its zeta potential of a particle, which might impact stability, interactions with excipients or with co-solutes. If you have a vaccine which is multivalent and you have interactions between the various antigens in the vaccine. And so light scattering and its variants do address all of these questions and provide a fairly comprehensive solution to this understanding. For today, we're going to focus on high throughput aspects of screening, which are mostly relevant to phase number 1, which is looking at a large variety of candidates or conditions and selecting out those which are most promising to continue on to additional studies. And so there are 3 essential flavors of light scattering: static light scattering or multi-angle light scattering, and it measures from first principles the molar mass of the molecules. You can also measure size of macromolecules and nanoparticles, measure of size known as the root mean square radius, or RG, and it can also measure from first interactions, from first principles, the interactions between molecules to get, say, equilibrium dissociation constant, KD, or a thermodynamic interaction measure known as B22, the second virial coefficient. Dynamic light scattering, on the other hand, measures from first principles the translational diffusion coefficient of the molecules, that is their diffusion due to Brownian motion in the solution. And from that you can derive a measure of size known as the hydrodynamic radius, RH. This can also give you, through the concentration dependence of RH or of DT, a measure of interactions between the molecules known as the diffusion interaction parameter. It's known in certain circles as KD, but I prefer to call it D1 in order not to confuse with other KDs such as the dissociation constant. And phase analysis light scattering is the third form of analytical light scattering, which measures the electrophoretic mobility, from which we can derive the zeta potential in solution and the effective molecular charge or the isoelectric point, other properties that are related to charge on the molecule. So if you're interested in learning more about these techniques, I do recommend that you visit our website, look at the theory section, listen to some webinars, and learn more about the solutions that light scattering provides. As I mentioned, for today I'm going to focus on high throughput screening, which is accomplished with this instrument, which is the Dyna Plate Reader 2, which measures dynamic light scattering directly in microwell plates. And as you can see in our schematic over here, we illuminate the well from below using a laser, and the scattered light is detected in the fiber optic cable. And so the laser and cable move around from well to well, make their measurements, and give you basically information on every well that contains sample in the plate. And so the Dynapro measures dynamic light scattering in standard microwell plates. These are industry standard plates, 96, 384, even 1536 well plates, so that you can measure dozens to hundreds to even thousands of samples per day. The range of size that the instrument can measure is between 0.5 and 1,000 nanometers. And its sensitivity for IgG, for instance, is 12.5 micrograms per milliliter. The sensitivity of dynamic light scattering in general depends on the molar mass of the sample. So if you have a smaller protein, for instance, lysozyme, which has a molar mass which is 10 times lower than that of IgG, its sensitivity will be 10 times higher or 10 times lower, meaning 125 micrograms per milliliter. On the other hand, if you're measuring larger particles like liposomes, the sensitivity will be very, very high, and you really don't need to have too many in solution or to get good measurements. The minimum sample volumes are stated here as well, and so if you're using the 1536 well plate, you can use as little as 4 microliters of sample at the concentrations mentioned here. The Dynapro offers temperature control, so it will do ramps from 4 degrees C up to 85 C, or you can do a ramp and hold and measure the rate of aggregation at a given temperature. It does offer automated attenuation, and the reason it does that is because when you go to one well, you might have a very clear solution which scatters very little light, and it needs to be at high gain in order to measure that effectively. And the neighboring well might have lots of large aggregates or large particles, in which case it scatters a lot of light and would overwhelm the highly sensitive avalanche photodiode without this automated attenuation. And so the implementation of auto attenuation allows you to measure a very wide range of concentrations and particle sizes. If you're interested in interfacing the Dynapro to a robotic liquid handling system so that you can automatically load plates in a robotic pipetter, load them into the instrument, tell it to go, acquire the measurements, and then pull that out and put in the next plate, there is a software interface that allows you to do that, and this has been utilized by a few customers in order to more fully automate their measurements of dynamic light scattering. And the Dynapro also offers a built-in camera, which will take an image of each well from below, and you can look at those and determine if there's precipitation in the well, or that it wasn't loaded properly and you have bubbles. Bubbles do impact dynamic light scattering and give you erroneous results. So if something looks funny, you can always go back, look at the image, and determine if it was a good measurement or not. So I'll begin this webinar after that brief introduction with more about the basics of dynamic light scattering and what it can measure. And then I'll go on to give a variety of applications for biotherapeutics and nanotherapeutics. For biologics, there'll be a little bit more, covering aggregation and stability, viscosity as well. And in terms of nanoparticles, a range going from gene-delivered nanoparticles through virus-like particles and to small molecule therapeutics, which are milled into nanoparticles as well, and we'll see how how dynamic light scattering plate reader can help accomplish high throughput process development. So imagine that you have particles in solution. What happens to the scattered light? Well, each of those particles will scatter a light wave, and you can see that depending on the relative position of the particles in solution, as I've shown here, the light waves may arrive at the detector in phase or out of phase. And if they arrive in phase, then they interfere constructively, and you'll have a high signal on the detector. If they interfere out of phase, then that gives you a low signal on the detector, and you'll have a variation. So as the particles jitter around in solution, they'll go from constructive to destructive interference, and the detector will go from a high signal to a low signal. Now, the rate of fluctuation will depend on the rate of diffusion of the molecules in solution, and that is the Brownian motion. So you can see that larger objects, such as this 40 nanometer particle, will diffuse relatively slowly and cause relatively slow fluctuations in the light intensity, whereas smaller particles, for instance this 4 nanometer particle, will diffuse around more rapidly and give rise to more rapid fluctuations. So you can see that here in a brief video. And what we do with that information is we measure the light intensity fluctuations, perform a mathematical operation known as autocorrelation analysis, and that takes those fluctuations, converts them into a rate of diffusion or diffusion coefficient. And then with additional information like temperature and viscosity, we can extract a value known as RH, the hydrodynamic radius. And RH is the radius of a sphere that diffuses at the same rate as our molecules diffuse. That can also be analyzed to extract an effective size distribution. And so you can see here a size distribution determined by dynamic light scattering. You can see that it's not very high resolution. So even though we show here, it looks like we have some particles at 2 nanometers and some particles at 4 nanometers and 6 and 8 and 10, it's not accurate. It's not a very high resolution technique. And you can see that the scale for the radius here is logarithmic, varying by several orders of magnitude. And so the size distribution is not highly quantitative, but it is pretty robust, and we'll discuss soon how robust a size distribution you can expect, and also what you can do in order to obtain higher resolution size distributions. The basic information that we get from the autocorrelation transform looks like this. So we see a decaying curve with a time scale, and this is fit to a Gaussian to extract an average radius, in this case 3.6 nanometers. And in fact, we can also determine a width around that radius. So, and that is converted into this distribution. So we see an average width here, 3.6, and an average value of 3.6, a little bit of a width for that distribution. If we have spheres of a different size, if we have particles which are larger, this will decay more slowly, looking at timescales which are longer, and it'll give us another peak in the size distribution here with a average value which is higher and its width as well. Now, if both particles are present in solution, then we get this autocorrelation curve over here, which actually combines information from both particles, and it will give us this dual peak here, each of which has its own average value and its own width. Now, the dynamic light scattering is very sensitive to the presence of large aggregates, and that's because the light scattering intensity is proportional to the molar mass of the molecules. So if we have a lot of small molecules, for instance BSA, and a very few large aggregates or larger molecule or larger particles such as these polystyrene spheres, the scattered intensity from the larger particles will show up very clearly, as it does over here. So if we have a solution which includes just one size of molecule or particle, that will give us a distribution with a single peak, and we call that monomodal. There is a measurement of polydispersity. So if there are some aggregates, say for instance dimers, trimers, we'll see a broadening of that peak, but we won't see a resolution in this distribution of dimers and trimers. So the dynamic light scattering analysis does not give us high resolution separation of oligomers. It just gives a broadening of the peak, and so we can get the average value of the peak and the width known as the polydispersity. Now, if there are two populations present which have very large differences in size, those may show up as distinct modes in the distribution. In particular, if the difference in size is a factor of at least 3 to 5 times in radius, then we can distinguish a second mode. And each of those modes has its average value, and it also has polydispersity in case there are inhomogeneous populations. For instance, there are aggregates, dimers and trimers of the smaller particle, or some variations in size, as you see here, of the large particle. And so in the complete regularization analysis, where we see the size distribution, we'll get one or more peaks. Each of those peaks has an average value and a polydispersity. And those can be tracked, for instance, as aggregation proceeds or some other form of degradation proceeds. We can see changes in those values of the average and the polydispersity. So to summarize what we have looked at so far in terms of the basics of batch dynamic light scattering, and by batch dynamic light scattering we mean in microwells or in cuvettes with no preliminary separation by size or some other property. It's a low resolution method in terms of size distributions. You can't see dimers or trimers as individual species. In fact, a factor of 3 to 5 in radius is required to resolve multiple peaks or multiple modes as we normally call them. On the other hand, the precision and repeatability of the average size of each of those peaks is about 1 to 2% if you do it right. So if you do these measurements very carefully and correctly, you can get pretty good precision and repeatability. Now, how do you know if you're doing it correctly? Well, for that, I recommend attending Light Scattering University, which is the training course on dynamic light scattering that Y Technology offers to each of its customers who purchases an instrument. Even though it is a low resolution method, batch dynamic light scattering does offer a broad size range. And so with the plate reader, you can measure from 0.5 to 1,000 nanometers. And if you're using glass cells or high quality quartz cuvettes, in fact, you can measure an even larger range depending on the particular instrument, radii from 0.2 nanometer and up to 5,000 nanometers. So it does cover a very large range and is effective for looking at populations which have a broad range of sizes. Dynamic light scattering in batch mode is pretty fast. So it only takes 5 to 90 seconds per sample, depending on size range and sample complexity. If it's a very simple sample, say a protein without large aggregates, it might just take a few seconds to make a good measurement. On the other hand, if the sample is complex and incorporates multiple modes and large sizes, then it might take a little longer to make a good measurement, but still pretty quick. It doesn't take very much sample. With as little as 4 microliters using the 1536 microwell plate. More typically, people use the 384 well plate, and there it takes about 20 microliters per sample. If you use a cuvette, Wyatt also offers the Dyna NanoStar dynamic light scattering instrument, which uses cuvettes, and there you can use as little as 1.25 microliters. So it is quite parsimonious in terms of sample requirement. And dynamic light scattering is quite sensitive to large aggregates or other particles. So if you're doing, say, a protein analysis and you are looking out for those large aggregates, it will show them up very clearly, even if you have small quantities, even if they're a very small part of the total mass of the sample. Now, if you're looking for higher resolution, you do in fact want to distinguish monomer, dimer, trimer, and maybe some other species which are not a factor of 3 to 5 in radius between them. You can use an size-based separation technique such as size exclusion chromatography or field fill fractionation upstream of the dynamic light scattering detector, and using a flow cell, a detector with a flow cell, run the separated sample so that each species is in this flow cell at a particular time. You make its measurement. Again, the precision is quite good, a couple %. And by that means, you can in fact get a pretty high resolution technique. Now, obviously, the dynamic light scattering plate reader doesn't support a flow-through cell, but we do offer other instruments that include a flow-through dynamic light scattering cell. Okay, so the first set of applications I'd like to explore are those related to proteins. Just the first thing that you do with a protein. What's the quality of the solution? Measure the size and the aggregate content. And you can do that with a plate reader to look at a large variety of conditions, of candidates, of formulation conditions or process conditions, along with multiple replicates for confidence and robustness in your measurements, and of course controls as well, which you need in any good scientific experiment. The Dynamics software, which operates the plate reader, will then color code for you the output of the measurements in this heat map, such as shown here. And so each color will correspond to a particular result. For instance, here, red corresponds to nicely monomodal, a narrow mode without aggregates. Blue will correspond to some aggregation, some increase in the width due to the polydispersity measurement, and perhaps some larger aggregates as we show here. And the black corresponds to very poor solution conditions, for instance, precipitation or very, very severe aggregation. Doesn't take long to run this entire set of measurements, 384 wells at 10 seconds per well, and the whole thing is done in an hour and a half. And so you can load up your sample, pop it into the instrument, click go, come back an hour and a half later, and all this information is here for you. A quick visual glance at the spectral view, which is the heat map, to see which types of formulations and conditions and candidates were good under those conditions, and which were aggregated and not actually suitable for continued measurement. And here's another example of doing the same thing in a 96 well plate. Here we're looking at a little bit higher resolution, so the measurement per well was a little bit longer, but all in all, 30 seconds per well. The whole thing was done in less than 45 minutes. And you can see here that how the color coding was carried out. Again, red corresponding to a nice sharp monomodal distribution, blue corresponding to some aggregates, and black corresponding to very heavy aggregation. And in this example, we're looking at a monoclonal antibody, which was formulated under two possible conditions, with or without 2% sucrose. And then looking at it before lyophilization, the material is then lyophilized and reconstituted, and we come back to the dynamic light scattering to see what types of aggregates formed. So before lyophilization, you can see that we have some pretty nice sharp distributions here. This is considered nice and monomodal and relatively sharp, and you can see there are not many aggregates in either one of them. The sucrose does have a little bit of increased width, and that's probably due to there's a little bit of interference from the sucrose because it can form some small nanoparticles. After reconstitution, though, we do see the addition of these larger aggregates, and in fact, the non-sucrose containing solution had some very large aggregates shown over here, and the sucrose containing condition just had this range of aggregates. So it was not quite as bad as the non-sucrose containing condition. The graph that is shown here is the size distribution according to intensity. So on the y-axis we have percent intensity, and we can see that we have a lot of intensity or a fairly large amount of intensity for the monomer, and smaller amounts of intensity for the aggregate. As I mentioned earlier, dynamic light scattering is extremely sensitive to aggregates, and even though there's appreciable intensity here, it doesn't come from a large mass of aggregates. The software allows you to convert this to what's known as percent mass graph, and in the percent mass distribution, you can see that the aggregates here are almost negligible. And so if we quantify the total amount of mass in the monomer peak, which is shown here, we can see that before lyophilization, 100% of the mass was in the peak number 1, which is this one over here, the monomers. And after lyophilization, 99.6% was still in that peak. For the sucrose, 98.8% of the mass was still in that peak, and only 1.2% of the mass went into the larger aggregates. And so don't be fooled when you see a very large peak corresponding to large aggregates of particles. They're there, and their presence is making itself known because they scatter a lot of light. But in terms of total mass, Turns out that in this case, they actually had very little mass. Now, how can you apply this same dynamic light scattering rule applied to characterizing size and aggregation of nanoparticles? So this is an interesting slide which compares different ways of measuring the size of a virus-like particle. So there are multiple techniques. The first one is X-ray diffraction, and it gave a value of 50 nanometers. TEM gave us a value of 33 nanometers, and it also gave this very highly detailed distribution of sizes. Dynamic light scattering gave a value of 52 nanometers, and it gave this low resolution distribution of sizes as well. So why do we get different values and what's the meaning? Notice that there is a distribution of sizes, and each one of these techniques gives you a different form of weighting. So XRD weights by the mass. Okay, so it's the weight averaged diameter, which is 50 nanometers. TEM gives you a number average value, and so the number average diameter is 33 nanometers. Dynamic light scattering gives an effective Z average value, meaning that it's weighted by the square of the molar mass. Now this is not extremely rigorous. It's not rigorously a Z average diameter, but it's reasonably close to a Z average diameter. We won't go into details, mathematical details, but that's approximately what we get. And we can see that it's about the same. And so that what that tells you is that at the high end of the distribution, there's the weight average and Z average about the same. Z average doesn't really see these smaller particles down here, which don't scatter a lot of light. And each of these gives you information which is important and relevant, and it's with different advantages and disadvantages. TEM is a pretty involved measurement. You have to prepare your sample, you have to put on a slide. There are a lot of things that need to be done. DLS is a pretty quick measurement. So it just takes a few seconds or a minute, and you get this information with very little sample preparation or none. Just pop it into the cuvette or into the well plate, and you'll get your information. And XRD also has its own requirements and effort involved. So it's nice to start with something rough, like dynamic light scattering. Rough, not rough, but I'd say quick and easy. Get a good measurement, and then proceed on to the others for more detailed measurements. Next example I'd like to look at for nanoparticles is titanium dioxide nanoparticles, which are being tested for toxicity to cell culture media. And one of the issues that you come across in making this measurement is that the particles need to be dispersed on their own. They don't disperse very well in all the cell culture media. They might clump up, and then it's not really a good measurement of toxicity. And so the folks at UCLA who provided these data we're looking at different dispersion agents, which included BSA at 2 mg per mil, and fetal bovine serum at a few different concentrations, 0.5, 1, 2, and 5% weight by volume. And for each of these conditions, including a control which had no dispersing agent, and 6 different media, including a control, which no media, just water, and 3 replicates for each of those. That comes out to over 100 different conditions. They populated the plate and loaded it up into the Dyna Pro plate reader, came back a little later and had all the results in front of them in the spectral view, the good, the bad, and the ugly. Those conditions shown here in red, which were reasonable. So red showed that an RH of 160 nanometers, which is basically individual titanium dioxide particles. The blue corresponds to slightly aggregated particles, and then black means heavily aggregated particles. And it's all here right in front of you. You can quickly identify the correct conditions. In this example, I'd like to look at some interesting work done at Florida State University, where the idea was to take a 900-bit pair gene, which encodes for a DS-red express fluorescent protein, and assemble it on the surface of a gold nanoparticle, and determine the best conditions for that assembly that will result in non-aggregating nanoparticles. So again, you could load up the plate with all the different conditions and come back and have all these nice graphs which show you the size of the free nanoparticle here, which was about 7 or 8 nanometers. If the gene is bound non-specifically to the gold, so it kind of wraps around the nanoparticle and is not hanging off, it gives you a relatively small size, which is only a little over 10 nanometers in radius. On the other hand, if the gene is bound specifically to the particle, it's attached at one end and the rest is hanging off, and that can lead to both a large size and interactions between adjacent nanoparticle gene constructs, which lead to aggregation. That leads to a much larger size here, which is on the order of 50, 60 nanometers. And then here's the size of the free gene, which is measured as well. It's because it is a linear construct which doesn't fold nicely like a protein, it gives you a pretty large size. So again, make all the preparations, put them into the microwell plate, and here you come back with the answers in a very short time. From basic characterization, I'd like to go on to stability measurements. And there are a few different ways of stressing particles and assessing their stability, assessing their propensity for aggregation as well. One of them is known as ramp and hold in terms of temperature. So you place your solution at an elevated temperature and assess the rate of aggregation that's caused by that elevated temperature. That's particularly important for proteins and other types of molecules which under temperature may unfold and aggregate. In this case, the basic proteins of the VLPs were beginning to unfold and disassemble and cross-aggregate. And you can see that depending on the particular type of mutant that was used, there are different rates of aggregation. They all start out for the first few minutes without aggregation, but the first C74S starts aggregating fairly rapidly. You can see that it's going up to very large sizes, from just a few nanometers up into the hundreds within a few minutes, whereas some of the other mutants, like the C74S and D9D1N, took longer and aggregated much more slowly, and therefore can be considered more stable, at least in terms of thermal stability. You can do assessment of aggregation under different formulation conditions, and here the group at the Middleburg group in Australia loaded up the plate with different types of excipients, so mannitol, trehalose, sorbitol, sucrose, and also with and without polysorbate, different conditions. Here in the graph you see the results in terms of the temperature onset of aggregation. So what they do is they load the plate, start ramping the temperature, and in the course of the temperature ramp, scan the plate and determine the average size and polydispersity at successive temperature points, and determine from that the temperature where it begins to aggregate. That's the aggregate, the onset temperature. And you can see that temperature varies depending on the type of excipient which was used, sorbitol, sucrose, trehalose, the amount of excipient used, so here's the percent weight per volume, and somewhere in there's also the information on with without polysorbate. So again, it's a very easy to carry this out. You perform all the different mixtures of excipients and polysorbate separately, load them into the plate, load the plate in the plate reader, and within a short amount of time you have all this information back in terms of assessing the thermal stability of each of these different formulations. Another type of drug delivery molecule or particle which is in common use is polymer micelles, and these also have temperature-based properties. These properties tend to be tuned by the particular type of polymer that is being used. And so it's important to assess the temperature behavior of the micelle formation. And typically that's done by fluorescence in, for instance, a qPCR. It's done in a qPCR by fluorescence because it's easy. And you can quickly load up the qPCR plate, do the temperature ramp, and figure out what's going on. The problem with that is that fluorescence is not a direct quantitative physical property of the molecule. It tells you that something is going on when it changes, and you can assume that change in fluorescence does correspond to, say, micelle formation or dissociation. But when you do it with dynamic light scattering, it actually tells you directly that the size is changing, and that size corresponds to the formation of micelle. And so here, in the course of this temperature ramp, we can see the variation up to about 40 C. It remains unimeric, and then at about 41, things convert into a micelle. And in fact, right around here, right about 41, 42, you can see that both species are present in equilibrium, the unimer and the micelle. And then around 50, there's an additional transition that goes on, which is an aggregation transition. And so the size is going up to about 1,500 nanometers, whereas the micelle size is only about, is a little less than 30 nanometers in radius. And so the dynamic light scattering result really gives you a direct measurement of what's going on, and in that sense it's more useful and more quantitative than a fluorescence measurement of CMC and CMT. Now, if you want to measure CMC, you're going to do a concentration series, and if you want to measure CMT, you're going to do a temperature ramp. In the plate reader, you can combine all these together. So you can load the Dyna Pro with, for each formulation or each type of polymer that you're assessing, a concentration series to look at CMC, and take that concentration series and run a temperature ramp, and simultaneously you'll get all the information about CMC and CMT in a single run. In this example, I'd like to look at the characterization of a production process for nanoparticles, where dynamic light scattering high-throughput screening also plays an important role. And this is work that was done at Merck by Dennis Lung and his colleagues. And what they're doing is they're taking a small molecule drug and converting it to nanoparticle form in order to have higher bioavailability. The normal milling process is very long and extended and not high throughput at all. And so they adopted a resonant acoustic nanomilling process, which actually works in 96 well plates. So you load up chunks of the drug and some particles which are used to affect the nanomilling. You also need to put in a type of surfactant which binds to the surface of the nanoparticles and prevents them from aggregating. And so you need to determine the right conditions in terms of which surfactant to use, what is the right ratio of surfactant to particle, and how long do you need to perform this nanomilling in order to get the right size of nanoparticle. And the desired size was about 400 nanometers. And you can see that, actually it's probably lower than that, it's maybe 200 nanometers. You can see that depending on the surfactant used, various types of tween, pluronic F127, pluronic F681, the amount used, so the ratio of surfactant to particle, you can get different final particle sizes, which ranged here from about 250 and up to 500, the smaller being better. And they also extracted at various points during the nanomilling sample. So they started with a 96 well plate, extracted sample into a 384 well plate, placed the 384 well plate into the Dyna Pro plate reader and made the measurements, then came back and did this 4 times. And so that fills the entire 384 well plate. And you can see that after 1 hour, there were not sufficient results, but after 3 hours, they in fact got the right size, and there was no need to go on to 6 or 24 hours where there was no change in the effective size. So this is a very high throughput and effective way of producing the nanomilled drug particles and evaluating them to make sure that you have the right result, all done in parallel in microwell plates. And this gives you a very, very rapid development cycle. So you can go back and try different things and go on to the next step, and you've completed this process development within a really short time. An important aspect of protein formulation is their conformational stability. Dynamic light scattering helps assess that both through the thermal stability and through other types of stability. But thermal behavior is considered a good proxy for conformational stability of a protein. And we see here the unfolding transition that lysozyme, for instance, undergoes in the course of a temperature ramp. Okay. Dynamic light scattering gives you information not just on the size at each temperature that you see here, but also the size distributions, which are shown here for a few of the points. For instance, the size distribution at low temperature, medium temperature during the course of the aggregate of the unfolding, and maximum temperature. And so you can determine, is there increase in polydispersity? Have aggregates formed? Has the average size changed? What's happened to your particle at each of those temperature points? Now, this type of measurement using dynamic light scattering is considered to be a very good proxy for conformational stability assessments. It typically agrees very well with calorimetry and circular dichroism. And interesting thing about lysozyme, which doesn't happen, unfortunately, for most of our therapeutic proteins, is that it is reversible upon cooling. So as you cool back down, it actually comes back down the curve and refolds. But there's additional information here, which is not shown in this graph, and I'll show you in the next graph. And that is the combination of size, which is measured here, okay, in the black points or the blue trace, and the dynamic light scattering count rate. Now, the count rate is a measure of static light scattering. And as I mentioned earlier, static light scattering is a measure of the molecular weight. Actually, to go a little deeper into theory, static light scattering intensity is proportional to the product of molecular weight and concentration. Now, in each of these wells, as we make the measurement and ramp the temperature, concentration is not changing. And so if we see that the count rate is not changing, that means that the molecular weight is not changing, meaning that there's no aggregation. So for lysozyme, besides the fact that we just have a small change in size during the unfolding, we don't see any large-scale aggregation, and we also find that it's reversible. We get additional confirmation that there's no aggregation because the molecular weight hasn't changed. Now, as I mentioned, that doesn't happen for most of our therapeutic proteins, but it is a really useful measurement to look at the count rate or the static light scattering in the course of these temperature ramps to verify and differentiate between unfolding and aggregation. Here's another example of using high-throughput screening in the dynamic light scattering plate reader for thermal stability, looking at some different formulations, each of which had multiple replicates, and again doing the temperature ramp, looking at the unfolding and aggregation. So if we were to do a reverse ramp, we would find if any of these are reversible and refold. Some additional information we see here is that at the end of the ramp, the sample is held at a fixed temperature of 70 C for some time, and the green shot up in terms of size, so there was more rapid aggregation, whereas the blue almost did not change in size, and so that in fact could be unfolding without additional aggregation. All that information is here. The results that we get from this type of temperature ramp do correspond very nicely to DSC, to calorimetry, and so this is a good technique which operates in high throughput and gives you additional information, which is the size and the distribution of sizes that result from the temperature stress. Similar to virus-like particles, another useful way of assessing the conformational stability of proteins is to hold them at an elevated temperature and assess the rate of aggregation. And we can see here a protein which within the first few minutes increased quite quickly in size. Not too large, it wasn't significant aggregation, but there was a rapid increase in size from a little under 4 nanometers up to almost 10 nanometers. And then after 50 minutes, it went to a different state where it was aggregating, but much more slowly. Interestingly, the same behavior was seen with fresh protein, which is the open squares, day 0, and with protein which had been sitting on the shelf for 13 days. And so that shelf at room temperature didn't really impact aggregation. There was no significant change. They behaved the same way. It did not aggregate separately, had the same size range in the beginning. And so that's a good way of assessing two types of stability at room temperature and then again at elevated temperature. Another way of assessing conformational stability is through chemical denaturation. There are certainly some issues with measuring stability at elevated temperature because processes that occur at elevated temperatures like 60 degrees aren't really relevant to room temperature. So you can look at the chemical denaturation, which takes place at room temperature and assesses the stability of the protein through the unfolding that occurs at different concentrations of a denaturant such as urea. And this work was contributed and published by some folks at Sanofi. And we see that, in fact, there's this increase of size shown in the change in hydrodynamic radius, which corresponds to unfolding. And there are three plateaus or three transitions that correspond to the three different domains which were unfolding. And so again, this type of measurement is typically done with fluorescence, intrinsic fluorescence usually, and it depends on the presence of the fluorophore, of assuming that the fluorescence actually does indicate a particular unfolding process, and hoping that you'll see it in each of the domains. There's no guarantee that it will be relevant for each of the domains, whereas a change in the size is a change in size. It's a physical change in the molecule which directly indicates unfolding. So that's why dynamic light scattering is really powerful for assessing conformational stability by chemical denaturation as well as thermal denaturation. Another aspect of stability of proteins and nanoparticles is their colloidal stability. And so briefly to indicate what that is, if you have two molecules in solution, they interact. They interact through what are known as colloidal interactions. The first type of interaction between them is simply hardcore repulsion, and that's because the molecules can't penetrate each other. So that's one type of repulsion. And if the molecules have a net charge, which is the same sign, they will repel each other. So these two molecules each have a net charge of +2, and therefore in solution they'll repel each other due to that net charge. In particular, if they're relatively far away from each other, primarily the interaction that they'll see will be that charge-charge interaction, and it will be repulsive. On the other hand, there are attractive interactions that arise as a result of charge. For one, the inhomogeneous charge distributions. So the red plus charges tend to be concentrated on one side of the molecule, here, and the blue negative charges are concentrated on the opposite side of the molecule. And now the molecules in solution will reorient in order to obtain the minimum free energy, which means that the pluses from one molecule like to be opposite the minuses from the other molecule, and when that happens, they attract each other. There are also surface dipoles, and there are other effects that lead to attraction, for instance, hydrophobic patches, hydrogen bonds, salt bridges, all the various things that happen on the process of proteins with which you're quite familiar, I'm sure, which lead to attraction. And so the net effect of all of these types of interactions is the colloidal interaction. In fact, it does depend on distance. So if the molecules are close to each other or far away from each other, they'll see different degrees of interaction. If they're oriented differently with respect to each other, they'll see different degrees of interaction. We quantify this type of interaction using a measure of protein-protein interactions known as the diffusion interaction parameter. And we measure the diffusion interaction parameter by measuring the diffusion coefficient as a function of concentration. The reason we do that is because the higher the concentration, the closer the molecules tend to be to each other, and the more they tend to interact. And what we do is typically load up a series of concentrations, for instance from 1 to 10 milligrams per milliliter, and measure the diffusion coefficient, which typically changes as a function of concentration. The slope of this line will give us the measure of D1, which is the diffusion interaction parameter, also known to others as K sub D. And D1 is related to a few different important parameters. One is A2, the second viral coefficient. Now A2 is a thermodynamic parameter which indicates interactions. It's really the one that we want to get at, which tells us if the molecules are in net, attractive, or repulsive. It's possible to measure A2 using static light scattering, but typically that involves a lot of sample and is not efficient for high throughput screening. So the D1 measurement, the diffusion interaction parameter, which is measured with dynamic light scattering, is amenable to high throughput screening, and it's something that we can do in the plate reader, and so it's used as an effective proxy. There are other parameters that go into the calculation of D1. They're not really important, but what is important is the trend. So both A2 and D1 have the same trend. If they're positive, or the more positive they are, the more repulsive the interaction tends to be. The more negative they are, the more attractive the interaction tends to be. So how can we utilize that information? Well, what we can do is we can load up a plate reader with with a protein under different conditions. For instance, different pH values, different excipients. We can do a temperature ramp to assess the thermal stability. And for each of those conditions, we can also load up a concentration series to assess the colloidal stability. And so here what we've done is we looked at one condition, which is pH 8.5, and we see that we have nice small molecules, basically monomeric, as we ramp the temperature up until about 56 degrees, at which point there is increase in size indicating aggregation. They're going up to hundreds of nanometers, so there's really significant aggregation. And then up here at about 74, 75 degrees, there's a secondary transition indicating an additional instability of perhaps a different domain of the molecule, which is now unfolding and causing additional aggregation. If we zoom in the area of the transition, we see that here, and we can look at the effect of concentration. So the blue diamonds tend to be the lowest concentration, and the gold circles are the highest concentration. And it appears that the onset temperature tends to shift with concentration. So the smaller molecules have the highest onset temperature, and the largest concentration has the lowest onset temperature. Okay, so that's one way of looking at this result, and we'll discuss another one later on. Now this is the same monoclonal antibody at a different pH, 9.5, and you can see that it has rather different behavior. It also goes through a temperature transition, but instead of going up to the hundreds of nanometers, it just goes up to about 15 to 25 nanometers in size, and so it's a much more controlled, I call it oligomerization rather than aggregation. It also has a secondary transition here at about 75. And again, we can zoom in. Now, when we see a transition like this, instead of specifying the onset, which is where the transition begins, we can also specify a midpoint temperature, which is more akin to what you would assess with, say, differential scanning calorimetry, the midpoint temperature. And those can be quantified as well for each of the different concentrations. And we see again that there's a little bit of a difference in the midpoint temperature depending on concentration. Additional information we can get from dynamic light scattering is what type of aggregates form. So if you look at, say, 80 degrees C, where we do have the different aggregation, we find that pH 9.5 gives us a nice small aggregate peak around 18.5 nanometers with no significantly large aggregates, whereas pH 8.5 has one mode focused around 73 nanometers and another mode which is centered around 1100 nanometers, essentially precipitation or close to precipitation of some very large aggregates. Now this is crashing out of solution. You might detect this crashing out of solution through a differential scanning calorimetry as a loss of signal. Dynamic light scattering tells you what's happening. This is not crashing out of solution, and so DSC would not tell you that you have aggregation, but dynamic light scattering gives you exactly what's going on. Additional information we get from dynamic light scattering is static light scattering. As I mentioned earlier, we can get the count rate, which is a measure of static light scattering, and because the contents of each of the wells has a fixed concentration, the change in count rate corresponds to a change in the effective molecular weight, or the average molecular weight. And you can see here that the average molecular weight changes by a factor of about 4 to 12 depending on the concentration. So that more concentrated solution forms larger oligomers. The less concentrated solution forms smaller oligomers. Let's get back to the diffusion interaction parameter. As I mentioned, in this example, we loaded a concentration series for each of the different pH values. And so from that concentration series, we can measure D1. And we see that for the pH 9.5, D1 is slightly negative. It's a value of minus 10. And it stays approximately at that value as the temperature increases up until about 52 degrees right here, where it starts plunging to a much lower value. When it reaches its minimum, that's where the effective RH that we measure actually starts increasing. So the onset of aggregation here corresponds to the minimum in the D1 value. And then once the aggregation occurs, D1 comes back up a little bit and then reaches some plateau right around here and then continues on. And this plateau more or less corresponds to the temperature where the aggregation stops occurring, it stops increasing. So it remains aggregated but does not increase in size. So what does that tell us? Well, it tells us that through D1, we can find an earlier onset of some change in the colloidal interactions at a lower temperature than is needed for aggregation, which tells us that something is going on. So the temperature is going some conformational change, which is not a significant change in size. It's perhaps revealing, just beginning to reveal a hotspot for aggregation without true unfolding. And that hotspot is leading to colloidal interaction between the molecules in solution. As the temperature increases, that hotspot is exposed more and more, still not undergoing major significant unfolding, until it reaches some significant threshold value here, where the hotspot is now fully exposed. The size increases, indicating that there's significant unfolding and eventually aggregation. Now, once the aggregation occurs, those hotspots from the adjacent molecules will stick to each other, and they'll now be hidden from the solution, and so therefore the molecules are less self-attractive because the hotspots have been hidden, and that causes the increase of D1. So by dynamic light scattering, we're getting information not just on the temperature for the onset, and not just on the degree of colloidal stability of a particular formulation, but we're also getting information on what happens around the thermal transition, what changes in the molecule are leading to different types of interactions between the molecule, and how those eventually impact the aggregation. I mentioned that you can measure static light scattering as well, and you can see here the count rate. And we can see here that the RH begins changing and increasing a little bit earlier than the change in count rate. And so that corresponds to unfolding, and then only later on do we actually have aggregation, which shows up as change in the count rate. So all that information is contained in this one dynamic light scattering run on the plate reader. Another important measurement for formulations of high concentration proteins is their viscosity. We can measure viscosity with dynamic light scattering, and we do that by using a probe particle with known size, typically something like a polystyrene latex bead, for instance, radius of 100 or 150 nanometers. If we know the size, which is RH here, and we measure the diffusion coefficient through dynamic light scattering, dt here, we can then extract the viscosity of the solution, and this is the viscosity of the protein solution. So we're ignoring light scattering from the protein. Actually, if this is done correctly, we don't see light scattering from the protein. It's overshadowed completely by the light scattering from the spiked-in probe particles. And we measure the effective diffusion coefficient of the probe particles, and that gives us we back out the viscosity of the solution. Is it a good measurement of viscosity? So that's been shown in various publications. For instance, this initial publication by Feng He and his colleagues who were at Amgen at the time, and they measured viscosity of protein solutions using dynamic light scattering in the plate reader and using a standard viscometer, macroscopic viscometer, and show that the values agree quite well, and also that this effectively distinguishes between two different buffers of high concentration protein, one of which leads to low viscosity to high concentration, 120, 130 mg per mil, and one of which corresponds to a much higher viscosity, which would not be suitable for processing and is not injectable. You can go beyond that. In fact, some of these authors from Amgen have published more advanced papers where they combine multiple assessments of a formulation to create phase diagrams such as these. For instance, at a given concentration and a given pH, how does the aggregation behave? How does the viscosity behave? And how does the temperature onset of aggregation behave? And so in this region, for instance, the criteria is that the unfolding temperature or the melting temperature is above 50 C. In this region, the criteria is that the viscosity is above 6 centipoise, and depending on the excipients that are included, there's different behavior. I believe that the white region actually is the region with appropriate values, and so you can determine those excipients which impact various measures of stability and goodness of formulation, and expand your design space to include the most appropriate values. You can do this on the plate reader, simultaneously measuring viscosity, measuring temperature onset of aggregation, measuring D1, the diffusion interaction parameter. You get all these at the same time. And so to summarize, high-throughput dynamic light scattering. It means that you can measure dozens to hundreds of samples in a well plate, just load them up and walk away. You'll determine the size, aggregation, and various measures of stability and propensity for aggregation. For multiple production formulation conditions, you do that with extra replicates without extra hassle. Just load up replicates side by side and you'll get all of them at the same time. Load up your controls, all that information will be included. You come back, you bring up the spectral view heat map, and you see which of those are suitable for continued study for formulation and process development. You get multiple measures of stability, which are orthogonal, the temperature of the onset temperature, the conformational stability with chemical denaturation, the diffusion interaction parameter for colloidal stability, and the type of aggregates that form. And all this can be done with very low sample consumption, down to 4 microliters per well. So high-throughput dynamic light scattering is great for enabling data-rich assays. You get all this information with the replicates and the controls so that you can explore a large space of formulation or process development, get sufficient information to be confident that you're choosing the right conditions with which to proceed. So with that, I'd like to thank you for your attention. If you're interested in more information, I invite you to visit our website, wyatt.com/DLS, which is an overview of DLS, and wyatt.com/HT-DLS will give you specific information and access to additional resources about the DynaPro plate reader, including additional webinars, and white papers and application notes and the brochure as well. You're also welcome to email me at dsom@wyatt.com. So thank you, and with that, I am ready to take on any questions.
Thank you very much, Dr. Sohm. We have a lot of questions, so let's get started. Peter asks, can this instrument detect IgG dimer versus monomer if you're looking at 300 kilodaltons versus 150 kilodaltons?
Right. So as I mentioned, it is not high resolution. If you have a solution with just monomer and another solution with just dimer, you will definitely see the difference between them in terms of their size. But if you have a mixture of monomer and dimer, what you will see is a little bit of a shift in the average radius and a little bit of broadening of that peak. And there's no way to establish definitively that it is a dimer versus trimer or some combination of dimer and trimer. You don't get distinct peaks.
All right. From Balaji, how do you automate measurements with the DLS system?
Okay, well, you load your solutions into the plate. The software allows you to set up a list of conditions, which sample is in which well, which buffer conditions, all the information you need, which pH, which salt concentration, all these things will be loaded in via the software. And then you tell it go. So depending on which type of measurements you need to make, if it's all room temperature and you're just looking for aggregation or just measuring the diffusion interaction parameter at room temperature, tell it go, come back, and it's pretty much done for you. And if you need to program a temperature ramp or some other ramp and hold, those are all programmed in. So you can set up that program, click go, and walk away.
Okay. The next question, is it best to use DLS or CG mols if you're measuring self-association and concentration?
Is more of a qualitative measurement of self-association, whereas CG mols will give you, CG mols is the first principles technique. So it will give you more thorough results. It will give you actual KDs. It will be able to distinguish between formation of multiple types of oligomers, but it does require more sample, and it is a slower technique.
I do want to thank you all for coming today on behalf of Cambridge Health Tech Institute and Wyatt Technology Corp. Again, thank you so much for coming. We look forward to seeing you at future CHI Global Web Symposia. Thank you.